Hello guys, this is Rishabh, a PMRF scholar from IIT Kanpur, and I'll be teaching you a very curious course, RNA biology. So we'll be learning very interesting aspects of the RNA world and how RNA is being used in our molecular system to perform different functions. So stay tuned for all the fun aspects of RNA through this course. And so today we'll be starting with our first lecture about the RNA biology course. So starting with the ribonucleic acid or the short form RNA. So, what is actually the ribonucleic acid? So, going by its name, ribose, ribo comes from the word the ribose sugar. So, it is a nucleic acid that contains the ribose sugar. So, ribose is a 5 carbon sugar. Then, nucleic comes from the word nucleotide or a word that belongs to the nucleus, and then acid because it's acidic in nature due to the presence of phosphate group. Okay, so here comes the meaning of the word ribonucleic acid. Moving on to its structure, so as we discussed just now, this is a base. This is the pentose sugar, and these are the phosphate groups. So these three components are collectively called as a nucleotide. So nucleotide consists of a sugar connected with the base as well as the phosphate groups. Okay, so this is the complete definition of a nucleotide, and these are the three components of a nucleotide to be starting. Now, moving on to a bit in the structure and starting with the nucleic acids. So, nucleic acids was discovered by a scientist called as Frederick Miescher. So, what he discovered was he discovered the nucleic acid from nuclei of pus cells. So, what basically he did was he took bandages from hospitals. Those bandages have pus cells. So he tried to isolate the nucleic acid that was actually negatively charged. Okay, so at that point of time, it was out of the knowledge of many historians. So that's why it was named as nuclein. Okay, so later, nuclein was found to contain basic proteins and phosphorus containing organic acid. Okay, that's now called as a nucleic acid. So nucleic acid is divided into two types. One is DNA and the other is RNA. So DNA is generally double stranded, and as the name suggests, it is deoxyribose nucleic acid. So the sugar is deoxyribose, and it consists of four bases: adenine, guanine, cytosine, and thymine. Okay, and this double stranded structure forms a helix through the complementary base pairing between A to T and G to C. That's how it forms a double stranded structure. Now, talking about the RNA, it's generally a single stranded structure, and as the name suggests, it contains the sugar ribose, and the four bases that are present here is A, G, C, and U. So here the T is replaced by U. And talking more about the different features of DNA or RNA, DNA is a bit stable structure and RNA is a bit unstable structure. How, we'll study in a couple of slides. And then we have something called as DNA mostly behaves as the genetic material. So it stores the information. But RNA is generally the used for transmission of information because it's not stable, so it cannot store the information. So that's why it is a form of transport of information from one mode to another. Okay, so these are some of the differences between the DNA and RNA. Now moving on to the basic structure of a nucleic acid, that is so nucleic acids are composed of nucleotides. Okay, so the nucleic acids are polynucleotides. That means various units, repetitive units of nucleotides are present in both the nucleic acids. So nucleic acid, so this is the unit of a nucleic acid. Now nucleotide consists of a ribose sugar that is joined with the base and the phosphate group. So this is called as a nucleotide. Okay. Then here comes one more thing. If we remove the phosphate group, so that forms a nucleoside, the base attached to the sugar molecule, that is called as the nucleoside. And if we attach phosphate to that group, that is called as nucleotide. Okay, now there are three components of a nucleotide. First is comes the sugar. So this is a pentose sugar, as you can see here. And we label this particular sugar because this helps us in labeling the directionality in terms of the nucleic acid. Okay, so here comes the first carbon. So we label, how we label this, we label this with the prime value at their superscript. So this is 1 prime, then this is the second prime carbon, third prime, fourth prime, and this is the fifth prime carbon. Okay, so here, what is the features of this particular sugar? I'm not going into the detail of the carbohydrates, but here it is beta anomer and D type of sugar that is present. Why D? Because in human system D is the only carbohydrate that is present. Okay, and L are the amino acids. So this is the basic of the isomerism. I'm not going into the detail in that particular field. So here we have two kinds of sugars. One is the ribose that is present in the ribonucleic acid. Another is the deoxyribose that is present in the deoxyribonucleic acid. So the in case of ribose, at the 2 prime OH, at the 2 prime carbon, the OH is present, while in the uh, DNA, the 2 deoxyribose. So here the 2 prime OH is not present, and here it is replaced by an H. So why this thing happened? This leads to the in evolution, when the nature needs a more stable molecule. So it has been said that DNA basically evolved from RNA only. So when nature found out that RNA is a more unstable molecule and it's a reactive molecule, so it tends to mutate. So then something happened to make this unstable structure to a bit stable one, so that it is less reactive as compared to the RNA. So that's why The nature removed the 2 prime OH so that in the continuing chain this OH do not behave as any of like do not have a reactive group. Okay, so this is how evolution happened from RNA to DNA. Now moving on to the next group, that is the bases, the important component. Okay, so we have two kinds of nitrogenous bases. So why these are called as nitrogenous bases? Because these are nitrogen containing compounds, like organic compounds, that the nitrogen is embedded in the ring structure, and these are basic in nature. That's why it is called as nitrogen bases. Two kinds of nitrogen bases are present. One is pyrimidenes and the other is purines. So purines are these ones that have a two ring kind of structure, and the pyrimidenes are single ring structure. Okay, so here this is the chemistry wala part. So I'm not going into that detail why they are purines and pyrimidenes, but we'll remember the numbering that is being followed here. Okay, so this numbering will help you in the nucleotide synthesis also. So in purines, the numbering starts from this particular end. Okay, so this is first, second, then this is the third N, fourth, fifth, sixth, and here comes the 7, 8, 9. So seventh and ninth is also an N. Okay, so this is the case with the purines. And in case of pyrimidenes, the number starts from this N. Okay, so this is 1, 2, 3, 4, 5, 6. Okay, now purines are of two types, adenine and guanine, because of their different structure, different functional groups present in this particular structure. So in case of adenine, we have six amino. At the sixth position, an amino group is present. So that's why it is also called as 6 amino purine. Okay. Then in guanine, we have an amino and an oxo group at the second and sixth position respectively. So this is 2 amino, 6 oxo group. Okay. Now moving on to the pyrimidine part. So pyrimidenes are divided into three types: cytosine, uracil, and thymine. So cytosine is 2 oxo, 4 amino, 2 oxo group, and the thymine has a methyl group here. Okay, and two oxo groups. In case of uracil, this methyl group is removed and only two oxo groups are present. So this is only present in RNA, while this is only present in DNA. Okay, so this was the story about the different nitrogenous bases. Now comes the last part, that is the phosphate group. So phosphate is generally attached to the 5 prime end of the ribose sugar, and it can be either mono, di, or tri. That means only one phosphate group, two or three phosphate groups are present. That's why it makes the nucleic acid as negatively charged molecules. Okay, so and the bases are connected with the first carbon of the sugar. And in case of pyrimidenes, the first N is used for making the bond with the sugar, and in case of purines and uh, purines are attached to the ninth N uh, of uh, of their ninth N to the first 1 prime carbon of the pentose sugar. Now moving on to the RNA world and how the thing started. So like first we'll go with the evolution wala part. So first, as the scientists suggest that a big bang happened, a large explosion that was 15 billion years ago. Then following this, after nearly many billion years ago, about 5 billion years ago, the solar system was formed, and then came the RNA world. Okay, So that means the development of RNA followed by the DNA. Then the first cells with DNA appeared. Then recently only the mammals appeared on Earth. So the RNA world suggests that there was a stage in evolution where RNA was the main molecule. That means it performs both genetic as well as catalytic roles. Okay, so that means the other name for it was self-replicating ribozymes. Okay, how? Genetic means that it was a genetic material, and for a genetic material to survive, there is a term that it must be self-replicating. Okay, and the catalytic role, like we have enzyme for the protein component of the catalysis, then the RNA component is called as a ribozyme that performs some of the functions. Okay, we'll see in detail a bit. Then that's why this RNA was called as a self-replicating ribozyme in that particular era. So a simplest example of this particular thing is ribosome, because it has both rRNA that performs some of the functions of the enzyme like peptide bond formation. Okay, so now coming back to the RNA world hypothesis that was given by Booz, Crick and Orgel, that states that RNA may have preceded proteins and catalyzed all reactions for survival and replication of last common ancestor. Okay, so that means before the origin of proteins as the enzyme molecules, there was RNA that performed the function of the proteins. And similarly, before the origin of DNA, RNA was the one that performs the function of a genetic material. Okay, so this was the basic RNA world hypothesis that was stated. Now, this was the again the timeline of the evolution, that how things happened. So first the formation of the earth, then stable atmosphere is formed, then the origin of life appeared in the water. Then we have pre-RNA world, that means the nucleotides and like basic components that make up the RNA. Then comes the RNA world, then followed by the protein evolution, and followed by the all the three DNA, RNA, and protein world. So they work together. That's why this is the latest one, that is the presence of all three compounds. Then we have the different origin of different cells, different organisms, depending upon the reducing environment of the earth. And in later we had the oxygen rich atmosphere, after which the evolution of eukaryotes happened. So these all belong to the archaea and prokaryotes group. Okay, so where did all this start? So it has been suggested by some of the people that basically life came from the outside of the earth. So one of the examples of this is Murchison meteorite. So this particular meteorite was uh, like had fallen from space in Australia around 1969. Okay, and it was considered to be a it belongs to the carbonaceous chondrite class. That means it contains various organic compounds. Okay, and these compounds are basically the compounds that made up a living system in today's world. Okay, so many various kind of amino acid, aliphatic aromatic hydrocarbons, fullerenes, carboxylic acid, hydrocarboxylic acid, purine and pyrimidines, alcohol. sulfonic and phosphonic acids were found when it was chemically tested. So this suggested that there is something in the outer space due to which the life came on Earth. Okay, so to test this hypothesis and to further conclude on some of the conclusions, then like experiment was performed in laboratory to basically replicate the Big Bang theory, or the explosion that happened around 15 billion years ago. So the experiment was performed by Miller and Urey, and what they did was they took a glass chamber in which they basically used electrodes for spark discharge. That was the explosion from different stars when they would have collided, and the gases that were present at that particular time, that is methane, ammonia, water and hydrogen. So these were the gases that were majorly present during that atmosphere, and it was done at a very high temperature. Okay, so what happened when the explosion would have happened at that point of time, due to the presence of these gases, spark and the temperature conditions, the some of the organic compounds would have formed in this particular in in water. So what they did was they used water to collect all the organic materials, because it's easy to test the water for various biomolecules. Sorry. So they collected these water droplets that was formed from the explosion, and after they collected, they took samples and tested for what are the molecules that will be present, that might be present. So they found out various amino acids, basic amino acids that were found during this particular time. Okay, and various other organic compounds were also present in that particular water, and these compounds were nearly have same composition that were present in the Murchison meteorite also, as you can see in this figure. So most of these things were similar to that of present in the Murchison meteorite. So this also suggested that the life that has been originated via explosion, that might be present in the outer space. Okay, so yes, this was the basic conclusion from various experiments that were done during that time. Now, what did the evolutionary theory says? So, unity of life. All living organisms are formed of same material and function according to the same principles. Okay, so they are formed from same biomolecules like carbohydrates, then nucleic acids, proteins. The basic constituency of these biomaterials are same, and they function on the same principles. Okay, then all the organisms are descended from a single common ancestor. So they originated from a single common ancestor that was named as progenome by the last kingdom classification given by the Carl Jung. And what does the life need? Genetic information, energy to survive, and to do catalysis. Okay, to how to convert this energy to a usable form. So These are the things that the life needs to for survival on this particular earth. So, what is basically the RNA world? Okay, that means during the explosion, complex organic molecules was produced by various reactions due to temperature, shock, and the different gases were present at that time, and followed by there was origin of the RNA. So first molecule that originated was RNA according to the RNA world hypothesis. And this RNA does the work of genetic material as well as protein enzymes. Okay, so this was the basic function of the RNA that was originated first. Then originated the proteins that might be from RNA or from other means. Okay, it is not clear yet, but these proteins now took over the functions of the ribozymes. Okay, and then originated the last and the most important molecule, that is DNA. So this particular thing took over the role of genetic material. So this was the last common ancestor that was there, that have DNA as the genetic material, protein as the catalytic molecule, and RNA to perform various other functions related to the protein or DNA or the transmission of information. Okay, so what are the properties of the RNA? So it's a single stranded molecule that have four bases, A, C, G, and U. Okay, and generally, like in lab environment, when we isolate the RNA from cells, so we take the concentration at 260 nanometer because of the presence of the bases. They like excite in that particular wavelength, and we check the 8 to 60 by 280 ratios for the presence of any protein component, that is generally like measured at 280 nanometer, and 8 to 60 by 230 to check for any of the salt contaminants. Okay, so this is the scenario with the RNA, and then it functions as a storage of information, like as a genetic material in case of various viruses like HIV, the commonest example that we said. Okay, so this is the second point, that it is a storage of information, and we'll see that how it's not a good molecule for a genetic material. Then it's for transmission of genetic information in the form of mRNA. So, as per the central dogma, DNA transfer its information to mRNA, and mRNA transfer its information to proteins that perform the function or the phenotypic response. Okay, so this was the thing. So it's a good molecule for transmission because it's highly reactive. Okay, and when this information is short time, so when DNA needs to form information in terms of protein, it tells to RNA that has a limited time. So until the proteins are formed, the RNA will be in the system. As soon as the proteins are formed, the RNA is degraded so that the information is maintained inside the cell only. Okay, and then it also helps in regulation of gene expression in the form of other RNAs that are non-coding. That means they do not produce any protein, but they regulates the gene expression in form of LNC RNA, microRNA, siRNA, and all the small RNAs that are being studied. It also has occupied the role in catalysis. So these catalytic molecules are called as ribozymes, And the famous example is peptide bond formation by the help of ribosomes. Okay, then we have cofactor for it's a cofactor for enzyme molecules like NAD, FAD. Okay, because it has a adenosine moiety, so that means it also belong to the RNA family. Then for energy storage, we have ATP. That's a major energy currency of the cell, and it helps in the transmission of energy from one form to another, and the utilization of the energy also, and in signal transduction in terms of CAMP and CGMP. So these were the properties of RNA and how it has been like covering all the steps inside the in the molecular cell. So this confirms the presence of RNA and the presence of the RNA void. Okay. Now we'll see some of the things in detail. So starting with the so like these belong to the evidences of the RNA void. So first thing is it is the storage of information. So it acts as a genetic material, but that too only in some of the viruses only, like tobacco mosaic virus or the HIV, or the latest one was the coronavirus that also had the RNA genome. So for a genetic material to be there, we'll discuss this in detail, but here we'll see some of the things in brief. So genetic material must be stable so that it can store the information, and secondly it must be like have some of the limits in the evolution. If it would be unstable, then it will mutate more, so that would not be good for evolution part. So that would create n number of possibilities for the proteins or the for the cells to grow up, so that would not be good for evolution. So how, so it must be stable and less mutable. Okay, so these were some of the features. And RNA being more unstable rather than being stable, it cannot store information for a long time. So it tends to be mutated very fast. Okay, so that's why the nature decided to not keep the RNA as a genetic material for higher organisms. Okay, that's why it is only being settled to some of the viruses like the tobacco mosaic virus and HIV. So when these viruses replicate more, they tend to accumulate mutations more. Okay, so why they accumulate mutations, as you have seen in coronavirus also. So with every replication cycle, it accumulates some of the mutations that leads to difficulty in forming their vaccines and all those stuff. Okay, now moving on to the next thing, the transmission of genetic material. So this is the how it is used as a transmission of genetic material is given by central dogma. So what does this say? DNA replicates on itself, being a genetic material. It has to replicate and produce its own copies. It transfers its information to the RNA, that is mRNA, through the process of transcription, and the mRNA moves its information in the form of proteins so that it shows the phenotypic response. So this is done with the help of the process called as translation. Now, when RNA would be a genome, okay, so how would it replicate? So in the primitive era, there was no DNA, But in the modern era, there was something called as reverse transcription. So what happened when mRNA is genome in some of the viruses like HIV? So they apply the path of the indirect replication. So first they form the DNA, they replicate, and then again it forms the RNA, and then the viral particles are formed. Okay, so the process of converting RNA to DNA, it is called as reverse transcription, and it was given by Tenim and Baltimore. So this was reverse central dogma because uh, it, the information is being passed through RNA to DNA. Okay, so this was the second component. Moving on to the regulation of gene expression. So earlier it was thought that only genetics control the phenotypic response. But later it was found that the environment is also involved in generating the phenotypic response. Okay, so how does environment control the phenotypic response? Is through epigenetic tags and the non-coding RNAs. Okay, so they control the gene expression at various levels, being in transcription or translation that we'll see in like the detail in some in future. So what they do is the environment basically transforms this particular thing so that the levels of expression regulates with the environment the cell is present. Okay, so here the non-coding RNAs, because here what this RNA or mRNA will be a coding RNA because it codes for a protein that yields a phenotypic response. But here some of the RNAs are non-coding RNAs that do not yield a protein but help in regulation of various processes. So these non-coding RNAs are long non-coding RNAs, that is LNC RNA. Then we have microRNA that have various targets inside the cell. So whenever this particular RNA is present, so it can target various genes that belong to the same nature or different nature also. And then we have siRNA that are very specific to genes. Okay, and these act at the 5 prime or 3 prime UTR regions. Okay, so what are these UTRs? We'll discuss later, but these regions are help in regulation of translation. So all these, the production of these are depend upon different genes, and these help in regulation of the translation process in various cells depending upon the environment. Okay, now moving on to the catalysis part. So this is the structure of the ribosome, and this is the prokaryotic ribosome, and this is the eukaryotic ribosome. So prokaryotic ribosome has two subunits. This whole is called as 70S. So it has two subunits. Large is 50S, small is 30S. And the 50S has two RNA, 5S and 23S, and the small one is 16S. So 16S has two roles. First, it is evolutionary conserved. So it can be used in phylogenetics, and secondly it helps in protein synthesis, so positioning of the small subunit on the mRNA. Then we have the 23s rRNA, that is basically the peptidyle transferase. Okay, so it helps in the formation of the peptide bond. Then coming back to the eukaryotic ribosome, it's 80S, so subunit 60S and 40S. It has four types: 5S, 5.8S, 28S, and 18S. So 18S has similar function to 16S. 28S and 5S has similar function to 23S, that is, it's a peptidyle transferase, and 5S has a similar function to 5S. Okay, so this is the basic scenario. This was the first example for catalysis, that is ribozyme. Now the second one is group 1 and 2 introns. So what are these? So introns are the non-coding regions inside the cell. And like in some of the regions there are group 1 and group 2 introns present that can excise or splice their on the by their own, so they do not need any external protein source so that they can splice them outside from the mRNA. Okay, so these introns are basically self-splicing introns, and they can remove itself from the mRNA to form a mature mRNA. And these have nucleic nucleus role also. Okay, so it tends to degrade some of the other RNAs that are a target site for these particular introns. Okay, that we'll see in a bit detail when we'll study the ribozyme structure. Okay, now moving on to the cofactor enzyme molecules. So we have a major form that is coenzyme A. It also has a adenosine moiety. So adenosine, we have a ribose sugar with adenine molecule and one phosphate group. Okay, sorry, two phosphate group. This was joined to the 3' bonds. So this is a major molecule in metabolism and helps in formation of various intermediates inside the metabolic pathways. And it also belongs to the RNA category because here this adenosine molecule is joined to various vitamins like pantothenate and cysteine. This is amino acid. So this proves that RNA was a major role player in the mechanisms also. That's why it formed different complexes inside the cell that helps in different areas of study. Okay, so here it's coenzyme A, then this is the NAD, nicotinamide adenine. So here we have also the adenosine moiety again. Then we have the FAD molecule. So FAD also have this adenosine moiety. So this like again tells us that RNA was the major contributor during RNA work. Okay, now we have the energy storage and signal transduction. So for energy storage we have ATP, that is the major energy currency of the cell. And we have like for energy transduction, ATP is the main molecule. Okay, so this is also an example that RNA was also used as a energy source when other molecules were not there. Okay, then same with the signal transduction. This is a CAMP molecule where it it is a secondary messenger to amplify the signal. Okay, so this is how the RNA has controlled nearly all domains in the molecular cell so that it will be able to complete all the reactions. And yes, now it has been distributed by various other biomolecules also, but at one point it was the only molecule that controlled every other function inside the cell. So this comes to the end of our first lecture. So this was a shorter one, and in the next in the next lectures we'll discuss various things regarding RNA, the genetic material, the synthesis, the ribozymes, and various other steps. So stay tuned, and if you have any questions, you can ask.